Welcome to the biology practicals. In these practicals, let us learn about gametogenesis. Aim of the experiment: to study the discrete stages of gametogenesis in mammalian testis and ovary under a compound microscope. Principle: gametogenesis is a process in which diploid or haploid precursor cells undergo division and differentiation to form haploid gametes in sexually reproducing organisms. It takes place in the gonads called testes in males and ovaries in females. Spermatogenesis is the process by which haploid spermatozoa develop from spermatogonial stem cells or germ cells in the seminiferous tubules of the testis. Oogenesis is the process of the production of egg cells that takes place in the ovaries of females. The spermatogenesis and oogenesis exhibit many differences and can be studied in a TS of the gonads. Requirements: permanent slides of TS of mammalian testis and TS of ovary and a compound microscope. Observation: TS of mammalian testis. Testis is bounded by germinal epithelium floored by a thick layer of fibrous tissue, the tunica albuginea. The testis consists of many seminiferous tubules embedded in the interstitial tissues. Various types of cells are present from outside to the lunar in the order of spermatogonia, spermatocytes, spermatids, spermatozoa, that is sperm. Between the germinal cells, the pyramidal shaped cells called Sertoli cells are present. A large number of sperms have their head embedded in the Sertoli cells. The Sertoli cells or sustentacular cells or nerve cells provide nutrition to the developing spermatids and sperm. Connective tissue stroma is present between the seminiferous tubules. It contains small groups of interstitial cells called Leydig cells. They secrete testosterone, a male sex hormone. TS of mammalian ovary. A mammalian ovary is a solid structure bounded by germinal epithelium followed by a thick layer of fibrous tissue called tunica albuginea. The ovary consists of outer cortex and inner medulla. The cortex consists of many oval bodies called ovarian follicles at various stages of development. The development of follicle is in the order of primary follicle, secondary follicle, tertiary follicle, Graafian follicle, corpus luteum, then into corpus albicans. A mature ovarian tertiary follicle of a mammal is called Graafian follicle. It is a fluid-filled structure within which an ovum develops prior to ovulation. It secretes a hormone called estrogen. The ruptured Graafian follicle develops into corpus luteum, which is characterized by a yellow mass of cells. It secretes the hormone progesterone. The degenerated corpus luteum is called corpus albicans, that is a white scar. When you watch the permanent slide under the microscopic view, the TS of mammalian testis looks like this. The TS of mammalian ovary looks like this.